Hi, my name is Fabian Soto and I'm an assistant professor at FIU in Miami, where I head the Computational Cognitive Neuroscience Lab. This is a pre-recorded presentation for the 2021 meeting of the Society for Mathematical Psychology. The title of this presentation is A Neurocomputational Account of the Mechanisms Underlying Face Perception Biases in Depression. People with depression show a bias to interpret ambiguous faces as expressing negative emotion and a general impairment in processing of emotional faces. There is little understanding of the mechanisms underlying such biases. Because there is evidence of higher level biases in depression, the simplest explanation is that face processing biases are decisional or cognitive in nature, so whether or not there are any perceptual mechanisms involved is an important question. Signal detection theory can be used to dissociate perceptual and decisional processes in perceptual tasks. Imagine that somebody has to indicate whether this face is showing a sad expression. This involves two steps. First, the perceptual system extracts sensory evidence that the face is showing a sad expression. This process is influenced by perceptual noise, so the sensory evidence changes randomly from trial to trial. Such noisy representations are captured by these normal distributions, one for the neutral face in red and one for the sad face in blue. The distance between the distribution means represents their perceptual discriminability. Second, the observer sets up a decision bound indicating how much sensory evidence is required to decide that the face is showing a sad expression. The lower this bound, the more bias exists to indicate that the face is sad. Signal detection theory allows one to separately quantify these perceptual and decisional factors. The extension of signal detection theory to multiple perceptual dimensions, known as general recognition theory, allows researchers to study the interactions between two or more dimensions. According to GRT, such interactions can happen at the perceptual and decisional levels. Imagine that now the face also varies in identity. Changes in identity can interact with emotion processing in two ways. First, they can affect how a face emotion is perceived. For example, focus on the two blue distributions representing different identities showing sadness. The area between the two distributions, shaded in blue, is a measure of their distance. It measures to what extent changes in identity produce changes in perceptual representations of sad faces. This is known as a violation of perceptual separability and essentially represents an inability to filter out information about identity when processing emotion. The second way in which processing of identity and emotion can interact is at the level of decision making. The threshold used to make decisions about sadness might change depending on face identity. This is represented by two different bounds. This interaction is known as a violation of decisional separability, and it can be measured by the distance between bounds. Here, we use signal detection modeling to dissociate perceptual and decisional mechanisms underlying face processing biases in depression. Each task involved 4 stimuli, which result from the combination of 2 identities and 2 emotions. In each trial, a combination of identity and emotion was presented and had to be identified through a specific response. The pattern of confusion errors was fitted to a GRT with individual differences model, or GRT-WIND, using maximum likelihood estimation in the R package GR-Tools. Measures of sensitivity, bias, and perceptual and decisional separability were computed directly from the estimated model parameters and compared between groups through their 95% confidence intervals, which were estimated using parametric bootstrapping. There were 3 different tasks in each session: neutral versus sad, neutral versus happy, and sad versus happy. The first and second sessions involved the same tasks using 2 different stimulus sets, which was done to check the generalizability of results across identities. The third session was identical to the first, but included stimuli that were made ambiguous by increasing similarity in both dimensions. All the results are presented in this table, which you can check by pausing the video or downloading a preprint from this web address. In summary, happiness was in general more difficult to detect by people with depression, and there was a general deficit of emotion discrimination with ambiguous stimuli. There were no effects of depression on perceptual separability of emotion or identity. Regarding decisional mechanisms, we found that people with depression used different decisional strategies than those shown by control participants, but that those differences show absolutely no consistency across stimulus sets. An advantage of detection theory is that it can be linked to face encoding models from the psychophysics and computational neuroscience literatures to formalize hypotheses about how face perception might be affected in depression. A large body of work supports a 2-channel opponent encoding system for face emotion, where a level of happiness is encoded by 2 neural channels represented here by logistic functions of different color. The red channel responds most strongly to happy expression, and its response decreases with less evidence of happiness in a face. The black channel responds most strongly to an anti-happy expression, which for simplicity we assume is the same as sadness. When a face is presented to this model, it outputs two channel responses, which include neural noise added during processing. Note that the happiness level is not explicitly represented in the channel responses. An estimate of how much happiness is shown in the stimulus must be computed from the channel's output. Because of channel noise, this happiness estimate is also noisy and represented by the purple distribution, just as assumed by signal detection theory. This means that distributions simulated from the model can be used to compute signal detection theory measures. In our model, channel noise is assumed to be Poisson, and people are assumed to optimally estimate the level of sadness and happiness in stimuli using maximum likelihood estimation. We assume that in depression, the channels encoding happiness and other rewarding emotions show suppressed activity, as shown by the orange arrow. Here's a simulation of the discriminability of happy and sad expressions against neutral as a function of expression intensity. More ambiguous expressions are closer to 0 in the middle of the scale. The simulation reproduces the asymmetrical results found in our study. In depression, there is a large drop in sensitivity to happy expressions throughout most values of intensity, whereas the same drop is found for sad faces only at lower, ambiguous levels of intensity. That is, the model explains deficits in the detection of sadness in ambiguous stimuli as an indirect result of suppressed representation of positive expressions. The model also predicts prior results that it was not explicitly designed to explain, like evidence of normal identification of unambiguous expressions. Here, as expression intensity increases, the gap between blue and red curves becomes smaller and eventually closes for happy expressions, as shown for sad expressions. A number of studies have used paradigms in which multiple faces are displayed simultaneously to determine whether people with depression show biased attention towards negative or away from positive expressions. This figure shows the sum of activity in the two channels produced by a variety of expressions in a negative-positive continuum. And here at the right, you can see the specific values expected for unambiguously sad, neutral, and happy faces. Assuming that attention is biased toward a face proportionally to the overall activity that it produces in the model's channels, differences in height between bars represent the relative ability of the stimuli to compete for attention if they were presented and displayed together. The model predicts that depression would be accompanied by a drop in the ability of happy faces to compete for attention, either against neutral or sad faces, as well as a smaller increase in the ability of sad faces to compete for attention against neutral faces. Several studies have confirmed such predictions. You can find more details about this study and simulations by downloading a preprint from this web address. The model just presented assumes that depression is accompanied by suppression of activity in visual channels encoding positive emotions. There are at least two mechanisms by which this suppressed channel sensitivity could be implemented. First, the channel could be selective to different visual information in people with and without depression. People with depression could sample information to detect positive emotions that is less optimal than that sampled by healthy controls. Thus, they will be less sensitive to relevant facial changes pointing toward a positive expression, and more sensitive to irrelevant facial changes that do not point towards such expressions. Second, the channel is selective to the same visual information in people with and without depression, but the actual output of the channel is suppressed. One way to start arbitrating between these two options is by estimating the visual information used by people with and without depression to detect facial expressions, and particularly positive facial expressions. For example, one common formalization of a visual channel in the psychophysics literature is the linear-nonlinear observer model. This model assumes that the channel takes an incoming stimulus and compares it against an internal representation via template matching, implemented through a dot product of the vector of pixel values in the incoming image and the vector of weights from a channel template. Internal noise is added to the value obtained for template matching, resulting in the random channel output described earlier. This perceptual evidence is compared against a threshold to obtain a final response that classifies the stimulus as matching or not matching the property represented by the template. Importantly, the template in the linear-nonlinear observer model can be estimated using classification image techniques, also known as reverse correlation. In this procedure, participants are presented with a neutral face combined with random noise images, and asked whether they see a target expression. Different noise patterns are presented in each trial. The observer reports seeing the expression in some of them, but not in others. Averaging the noise images identified as the target expression produces an estimate of the template used to solve the task. The estimated templates, which are usually called classification images, highlight areas of the face that provide task-relevant information. In our second study, we used reverse correlation to estimate templates used for identification of happiness and sadness in a group of 30 participants diagnosed with depression and a group of 30 healthy control participants. On each trial, participants were presented with one of 3 different base images of faces showing a neutral expression: identity 1, identity 2, and the average of the 2 identities. A random noise sample and its negative were superimposed on the base image and presented to the participants, who had to identify which of the 2 noisy stimuli showed a stronger emotional expression. In separate blocks, participants were asked to judge either happiness or sadness in the stimuli. This study produced a large amount of data that we are still in the process of analyzing. Here, we will present the results of some preliminary analysis of classification images obtained using the average identity as a baseline stimulus. These images show the classification images for happiness superimposed on the baseline stimulus. Note that the classification images are just the average of noise patterns classified as happy during the experiment. One can see that the reverse correlation procedure recovered templates from both groups that can be perceived as a happy expression. However, there are some differences regarding exactly what changes in brightness were categorized as happy by each group. Those differences are easier to appreciate by plotting brighter parts of the classification image in red and darker parts in blue, as shown here. The most important difference between groups is that people with depression require darker and more extended changes around the left commissure of the mouth to classify an image as showing happiness. Dark lines next to the mouth commissures are important in both groups, but in the control group, much subtler changes were perceived as showing happiness. We perform a cluster test based on Gaussian random field theory to determine areas of the classification images that were brighter or darker than expected by chance. As shown in this figure, the results confirm that classification images for people with depression were significantly dark around the left commissure, whereas classification images for controls were significantly light right below the left commissure. To determine whether these differences were themselves significant, we computed the difference between the classification images for both groups and performed a cluster test on the resulting difference image. As shown in this figure, there were multiple areas of the classification images that show significant group differences, among them the area around the left commissure mentioned earlier. In the encoding model presented earlier, we assumed that people with depression show suppressed activity of neural channels sensitive to positive facial expressions when compared to normal controls, but they show the same activity of neural channels sensitive to negative facial expressions. In the context of opponent channel coding of expression, this simple model is all that was required to explain the psychophysical data obtained in our first study. The classification images obtained here for sadness offer a test of this assumption. As before, the reverse correlation procedure recovered a template that can be perceived as a sad expression in both groups. However, again the patterns of darker and brighter areas seem to differ between groups. For example, only the control group seems to rely on lips turned down to detect sadness, and only the depression group seems to rely on eyebrow frowning. The results of a cluster test on the difference between classification images supports the hypothesis that the 2 groups relied on significantly different facial information to detect sadness. These results suggest that a revision of the model presented earlier is required. Changes in sensitivity to visual information seem to be found for the detection of both positive and negative emotions, although more work will be required to formalize a model that can account for the results of both studies. In summary, here we took a computational approach to understand the mechanisms underlying face perception biases in depression. In our first study using multidimensional signal detection theory modeling, we found that depression is accompanied by lower sensitivity to happiness in general and lower sensitivity to both happiness and sadness with ambiguous stimuli. We showed that suppressed response of neural channels encoding positive expressions in depression provides a formal account for those results and other results found in the literature. In our second study, we used reverse correlation to characterize the channel templates used to detect happiness and sadness in faces, and found that those templates differed between people with and without depression. We are currently working to formalize a model which can account for the results of the 2 studies presented here. The present work is a step forward toward a better understanding of the mechanisms underlying face perception biases in depression, and toward formalizing the assumptions behind cognitive theories of depression in the form of concrete, testable computational models. Face perception is a particularly advantageous area for such developments, as faces are complex stimuli with social significance, but they are also easy to manipulate and intensely studied in basic research. We hope that the approach taken here can be expanded to incorporate other biases found in depression.